Okay, good morning, everybody. Mike has put up some information on the Scantron. What you'll have to fill out on that Scantron on Friday. We provide Scantrons for you. This is gratis at UC Berkeley. Isn't that generous? Bring your SID number, please, and you have to enter that, and you have to enter your discussion section, not your laboratory section. So that's important. If we don't have that proper information on your Scantron, we can't grade your exam, obviously. Today is my last lecture, and I will have a few comments about that at the end, I think. Office hours today, 9 to 10 a.m. Then tonight, review 7 to 9 p.m. in the big lecture room in VLSB. I think that's 2050. And that's sort of your last chance to get your questions answered because I'm not going to be around Tuesday, Wednesday, and Thursday. So please come and ask questions. Mike will be here Wednesday and 8 to 9, and he will also answer questions. Now, they're still having trouble with the webcasts. What I recommend, or I suggest, is that you look at the fall 2010 lectures, which are archived on the Berkeley webcast site, and look at lectures that may be messed up currently. The lectures from 2010, they're not identical, but they're pretty similar. The photosynthesis lecture is certainly very similar, and the respiration lecture is very similar. So at least it's sort of a backup for you. Okay? Any questions? Remember, the exam is on Friday morning, and you have to look at B-Space to find out where you're supposed to go. You don't come. Some of you come to this room. I may be here. I don't know. That's the way it is in life. But most of you will be distributed around the campus, and you'll go to where your discussion section is supposed to go. Okay, so I want to briefly review what I spent an hour talking about on Friday, which was photosynthesis, the light reactions as we call them, and I've put up here the so-called non-cyclic electron transport part of the process. That's also shown in figure 126. And the point here is that we have two photosystems, PS2 and PS1, which cooperate in the transfer of electrons from water to NADP+. The electrons are passed through photosystem 2 to a quinone, plastoquinone, which is like ubiquinone, through a cytochrome complex, which has a B cytochrome, and something called cytochrome F, which is a C cytochrome, like complex 3 in mitochondria. So this portion of the chain is very mitochondrial looking. Then the electrons are passed to plastocyanin, a copper protein, not cytochrome C, through photosystem 1. There are a couple of electron carriers that function between photosystem 1 and NADP, but I haven't mentioned them, so you don't have to worry about them. I've shown on this figure as well that protons are liberated when water is oxidized, and protons are also moved across the membrane when this plastoquinone is being oxidized and reduced. You get a proton gradient in the light with the lumen becoming acidic, the stroma becoming acidic, and there is an enzyme in the membrane which is known as CFO and CF1, which is entirely analogous to the mitochondrial protein, which is known as FO and F1. This is the ATP synthase, and that enzyme can convert the gradient of protons into ATP. ATP and NADPH, the products, are formed in the stroma. And, of course, there's an additional product you should be aware of, and that's molecular oxygen. And I think most people realize that all of the oxygen on our planet, essentially all of the oxygen on our planet, comes from this process. I've put on here something about the light which activates these photosystems. I mentioned that when I talked about the reaction center complexes, that system 1 is preferentially activated by what we call far red light because it has a farther absorbing chlorophyll. System 2 by red light because it has a shorter wavelength chlorophyll as its reaction center. I was going to show you, but the projector doesn't work, some of the recent evidence and data related to oxygen evolution. I think you understand the significance of this reaction. It is absolutely critical for us because we use oxygen. It used to be called the black box of photosynthesis because we knew very little about it. But in recent years, there have been several groups that have crystallized photosystem 2, this entire complex, and they have elucidated a three-dimensional structure at a fairly high resolution. And what you find in the oxygen evolving complex are 4 manganese atoms. And there, I brought you a figure, but I can't show you the figure. There it is. I brought this in because a friend of mine did a lot of this work, Jim Barber, who's a professor in London at Imperial College. His lab elucidated one of these structures. You have 4 manganese atoms in a cluster, a cube. They're at the corners of a cube. You have 1 manganese that sort of sits outside, and there's actually a calcium in this structure as well. So there seems to be a structure for this oxygen evolving complex, but people still are working on the specific mechanism of how this reaction occurs. Very, very important reaction in the biosphere. Okay. Now, non-cyclic electron transport, that's what I've talked about, and photophosphorylation. The ATP that is synthesized in this process is called photophosphorylation, discovered in the 1950s by a professor on the Berkeley campus, Daniel Arnon, and he named it photophosphorylation to distinguish it from oxidative phosphorylation. So we have substrate level phosphorylation, oxidative phosphorylation, and now photophosphorylation. The original observations, however, on photophosphorylation were unusual. It turns out that if you take chloroplasts and you simply add ADP and phosphate and you turn the light on, they make ATP. They make ATP in the light. There's no oxygen involved. There's no NADPH formed. This process was called cyclic photophosphorylation. It's the synthesis only of ATP, and it only uses photosystem 1, this photochemical reaction center. There is no involvement of photosystem 2, and in fact, if you add this inhibitor that I talked about last time, and an inhibitor you'll use in the lab this week, DCMU, which blocks electrons coming out of photosystem 2, it does not affect this ATP formation. So it is independent of photosystem 2. How can this be? How can you just turn the light on and get ATP out of this? I didn't draw this because you have a figure that gives you some idea. I'm not going to go into all the detail because I have not identified all the components, but figure 127 shows that photosystem 1 can, under some conditions, instead of this electron going to NADP to form NADPH, this electron cycles around and is transferred. So here's the electron, transferred to plastoquinone, and then it uses the same portion of the non-cyclic pathway, but it is only involving photosystem 1, and it is making ATP by pumping protons, moving of protons across the membrane as plastoquinone gets oxidized and reduced. There are some of the specific electron carriers shown in this figure. That's not terribly important. But what you're doing in this system is you're simply taking the electron up and then recycling it around photosystem 1. If I had some interesting color, I could do this. Electron is going this way, and that way. That's the cyclic process. It does not involve photosystem 2. Now, it turns out there are conditions where the plant can actually uses this ATP. It's not frivolous ATP. For example, I will talk about in a couple of minutes carbon dioxide fixation, where 3 ATPs for every 2 NADPHs are required. It's not simply a one-to-one relationship, and the fact that there's an additional ATP requirement for carbon fixation has led some people to propose that it is the cyclic process of making ATP that's providing this extra ATP, because during the non-cyclic process you essentially get equivalent amounts of ATP and NADPH formed. Okay? Okay. Figure 128 is sort of a summary. I don't know why I put figure 128 in. I've talked about all of that. It shows you, again, making the proton gradient and the gradient being used to synthesize ATP. So what we're going to turn our attention to now is the second box of the 2 boxes on photosynthesis, the CO2 fixing reactions, or the Calvin cycle, as we call it. The Calvin cycle is called the Calvin cycle because the cycle was worked out by a Berkeley professor, Melvin Calvin, in the chemistry department, 1950s. And he worked in, I think, Lewis Hall. He taught organic chemistry for a number of years. He was not a renowned instructor, I've been told. He was on my qualifying exam back when I was a graduate student, which I suppose was an honor. Didn't ask any questions. But he worked out this cycle, and he received the Nobel Prize in 1954 for this. Nobel Prize in Chemistry, NP. You know, you've seen these parking places on campus, I hope. Nobel Prize parking places. If you get a Nobel Prize, you get a free parking place from the campus. Big deal. Okay, there were several things that happened in the late 1940s and early 50s that allowed the Calvin lab, and this was work done by a number of people in his lab, to elucidate this cycle. One is the development of techniques of paper chromatography for separating molecules, and you're going to use paper chromatography this week to separate pigments, but they were using the chromatography to separate different sugar molecules. And they also had a number of sugar molecules that were synthetically prepared so they could run these out under comparable conditions, and they could look at a chromatogram and say, this represents glucose or glyceraldehyde 3-phosphate or something like that. So they were able to do that. They also had a big advantage in that there were linkages between chemistry and LBL up on the hill, and LBL was one of the first places to start producing or getting radioactive carbon, C14 isotopes. So they were able to use radioactive carbon in these studies. And this is actually one of the reaction vessels they used. Anybody know what this is? No takers, huh? This is a lollipop. Okay? That's supposed to be funny. Nobody gets. Looks like a lollipop. Okay, what is this reaction vessel? There's a port up here where you could bubble CO2 into the system. They were using a green algae to start off with, and then they also did these experiments with spinach chloroplast. So they had a sealed vessel, they had a thin path length so light could go through, and this port was used to inject radioactive carbon, CO2, C14O2. The size of the stopcock is important because what they would do immediately after injecting C14O2 is they would spin this stopcock. It's frozen. It's been frozen for 40 years, I guess. And a sample would come out. And the sample would go into hot methanol. It would kill the organism, and it would stop the reaction. So by spinning that stopcock at suitable times, they would get a time course for the incorporation of CO2 into products. And that's what they were interested in. They were trying to identify the first products of photosynthesis. And in fact, they were successful. And if you look at figure 130, what one sees was that the first product of photosynthesis, after CO2 was, quote, fixed, was a compound that we talked about during glycolysis. It was a compound phosphoglyceric acid, a 3-carbon compound, C3. And what further studies showed was that this C3 compound was being formed by the reaction of a C5 sugar, ribulose bisphosphate, which is shown in figure 130. It's a ribose sugar, 2 phosphates on it, reacts with CO2, becomes a 6-carbon intermediate, and that compound is split in half to 2 C3 products. This is the most important reaction in this cycle. Okay? Again, I am not going through this cycle in detail, but this is the key enzyme that you have to understand the Calvin cycle. So we have ribulose bisphosphate plus CO2 forming PGA, catalyzed by the enzyme ribulose bisphosphate, and I'm only going to write this once, carboxylase, because this is adding a CO2 / oxygenase, and I'll talk about this later, what this means. Okay? If you can't read that, don't worry about it. It is shown in several places in the handout. This enzyme is known as Rubisco, R-U-B-I-S-C-O, because we don't want to say ribulose bisphosphate carboxylase / oxygenase. And this is the enzyme that actually fixes CO2. What happens after that reaction is there's a series of reactions that in many regards look like the reverse of glycolysis. What we're doing here is we're synthesizing a sugar, so you might expect reactions of the Calvin cycle are going to look like the degradative reactions of glycolysis, only in the reverse direction. So PGA, the first product, reacts with ATP to form 1,3-diphosphoglyceric acid, another compound we talked about in glycolysis. Then there's a reaction that requires NADPH. Remember, ATP and NADPH are required for CO2 fixation. And the product of that reaction is glyceraldehyde 3-phosphate. That should be an old friend because everybody by now has studied the details of that reaction from glycolysis. I'm going to stop here and start to count carbons because this is what's important. This is a cycle. In order for the cycle to work, you have to regenerate the starting material, just the way you have to regenerate the starting material in the Krebs cycle, oxaloacetic acid. Here, you have to regenerate ribulose bisphosphate so it can react with CO2. So we have 3 molecules of ribulose bisphosphate, which represents 15 carbons. We add 3 carbons more, so we have 6 phosphoglyceric acid molecules having 18 carbons. What we do after getting to glyceraldehyde 3-phosphate is we take one of these away because it's a product. That's a sugar. Remember, if you don't remember this, go back and look at my little table when I introduced carbohydrates. This is a triose sugar, glyceraldehyde 3-phosphate. The remaining 15 carbons, there are 15 carbons here, 3 carbons here. There are 5 C3 compounds. There are 15 carbons in glyceraldehyde 3-phosphate. They are used to regenerate the 15 carbons of ribulose bisphosphate. All of this is shown in figure 129, and that is sort of the level I want you to understand this process. The cycle is much more complicated than that. I actually do have a figure of the cycle. But, and I'm sure there's a figure of the cycle in the book. But I don't want you to get hung up on all of the steps that go on between the conversion of glyceraldehyde and ribulose bisphosphate. There's lots of biochemistry going on. There are all kinds of weird sugars. They may look weird, are being synthesized and converted into other sugars. There are 7 carbon sugars. There are 4 carbon sugars, on and on and on. I think that stuff is beyond me, and I think it's also beyond you. So that is not what's important in this particular process. What's important to realize is this is the first compound formed as carbon dioxide is fixed. It's converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is a sugar product. If I take 2 glyceraldehyde 3-phosphates, I can make a glucose molecule, for example. In addition to the 6 ATP here, at some point in the cycle, near the last step of the synthesis of this compound, there is an additional requirement of 3 molecules of ATP. So we have here 3 CO2s require 9 ATP, 6 NADPH to make 1 glyceraldehyde 3-phosphate. And here's where the extra ATP comes from, and presumably this, I can't say this ATP comes from the cyclic ATP synthesis versus this, but there is certainly more than a 1-to-1 relationship for the requirement of ATP in the Calvin cycle. So there's a second ATP utilizing step here, and there's a first ATP utilizing step here. Ribulose bisphosphate carboxylase, Rubisco, turns out to be the most prevalent protein on this planet. Every plant cell has it, and every plant cell has large amounts of it. Large amounts because this is a very sluggish enzyme. It doesn't work very well. It doesn't do its job very well. So the plant is compensated for poor activity by putting a lot of this enzyme in a chloroplast. All of these reactions occur in the stroma of the chloroplast, this region out here, where the ATP and NADPH are being made. So we've got stromal enzymes. None of them are on membranes. None of them are, no membrane involvement in this. Soluble enzymes. I should also mention that if you look at the complete cycle, you'll see there are lots and lots of different steps, and the Calvin lab worked very hard to show all of those enzymes were present. They predicted that the pathway, then they had to go look for the enzymes. And when you find the enzyme, you have to make sure its activity is sufficient to be involved in the cycle. And they did all of those things, and for that you get a Nobel Prize because it was a pretty big deal. Okay, now, not only is Rubisco a relatively inefficient enzyme, Rubisco is also, I like to say, a stupid enzyme. The name of the enzyme is ribulose bisphosphate carboxylase / oxygenase. Oxygenase because ribulose bisphosphate, Rubisco, I'm just going to say Rubisco, not only can react with CO2, but it can react with CO2. So we've got RBP, which plus CO2 gives PGA, and this is the carboxylase activity. But this enzyme can also react with O2, and when it reacts with O2, what happens is you get a C3 compound, which is PGA, but you get a C2 compound as a product because you only have 5 carbons. And the C2 compound is a compound known as phosphoglycolate. You don't have to know that. You can see, in this case, I'm getting 5 plus 1 goes to 6, and that gets split to C3. But in this case, I am getting C3, and I'm getting another compound which is not an intermediate of the Calvin cycle. This process results in this compound undergoing a number of biochemical processes and a number of different organelles within the plant. The net result of this is it's broken down and CO2 is actually released. So instead of fixing CO2, there are reactions in plants that release CO2. This is known as photorespiration, because respiration we can define as CO2 evolution through the Krebs cycle. But in this case, there's a respiration, there's a CO2 release, which is not happening by the Krebs cycle, it's happening by biochemistry on this C2 compound. What this means is the plant is not fixing as much CO2 as you would like it to fix. The job of the plant is to make carbohydrates, to make sugars. And I put down here, in fact, you take these C3 products, which is glyceraldehyde 3-phosphate, and you can make sucrose, and you can make starch. Starch is made in the chloroplast. Sucrose is made outside the chloroplast. You're going to look, you all know about starch now because you've had the lab from last week. The other difference between this photorespiration and true respiration is there is no ATP synthesized. So it is pretty much a wasteful process. And in some plants, it is a large component. 50% of the CO2 that could be fixed is actually lost as CO2 during photorespiration. Why does this enzyme do this? Well, obviously, CO2 and O2 are both substrates of the enzyme. This is not a competition. It is a competition for the active site by both compounds, but you get different products depending on whether you're using oxygen or carbon dioxide. And there have been enormous attempts and enormous amounts of money spent by agricultural companies to try to take Rubisco and genetically alter it so it no longer has this oxygenase activity. And that way, the enzyme would just fix twice as much CO2 as it normally did, because you would not have any losses due to photorespiration. And I'd say over the course of 30 or 40 years, DuPont has invested heavily in this. They haven't been able to. No one has yet been able to produce a carboxylase that does not have oxygenase activity. If you alter the active site of the enzyme a little bit to try to get rid of the oxygen reaction, you affect the carboxylase activity as well. So that has not been successful, although it certainly is a good idea, I guess I would say. However, there is, nature can take care of itself, I guess I would say. There are pathways of CO2 fixation in some plants which have little or no photorespiration. What do I mean by that? I mean, you can take plants, which are now known as C4 plants. They were discovered. These plants that are doing this with the oxygenase activity are known as C3 plants because PGA is the first product. There are plants which are known as C4 plants. You do the same experiment with a lollipop. You inject CO2. You don't find PGA as the first product. You find 4 carbon acids, like oxaloacetate is one we've talked about, this intermediate of the Krebs cycle, oxaloacetic acid. What's happening in C4 plants, okay, that was the first observation. The second observation was when people looked at these plants, C4 plants, they discovered that there were 2 kinds of photosynthetic cells in these plants. There were 2 kinds of chloroplasts. There were what are known as mesophyll chloroplasts and there were bundle sheath chloroplasts. And they knew these were chloroplasts because they were green, obviously. So there was no question about it. And what was discovered was that there was a second process in a C4 plant which allowed the CO2 to be fixed. It was not fixed into PGA. I refer you to figure 132. Figure 132 is an enzyme that is found in C4 plants, phosphoenolpyruvate carboxylase. It takes PEP, a compound that's actually in glycolysis, and it makes oxaloacetate. This enzyme exists only in mesophyll cells of the C4 plants. So in mesophyll cells, we have this enzyme, PEP carboxylase. The reason one sees C4 acids as the initial product is that PEP carboxylase is a better carboxylase than Rubisco. It works at lower CO2 concentrations, and it is more active. So when C4 plants are exposed to carbon dioxide, Rubisco does not initially react with the CO2. The CO2 is fixed by this enzyme PEP carboxylase. Well, what's the purpose of this? If you look at figure 133, the middle of the page, you can see the left-hand cell shows you there's CO2 coming into a mesophyll cell in a C4 plant, and it's fixing that CO2 into a 4-carbon compound. That 4-carbon compound is transported into a bundle sheath cell. It's moved across membranes and all that kind of stuff, and then it is decarboxylated. The CO2 in oxaloacetate is released, and that CO2 is refixed by Rubisco in a bundle sheath cell. What's the purpose of this? The purpose of this is that you can increase the CO2 concentration in the environment of Rubisco carrying out this reaction sequence such that it is much higher than simply related to the CO2 concentration of air. The oxygenase reaction and the carboxylase reaction basically compete with each other. As the oxygen concentration goes up, you get more oxygenase activity. As the CO2 concentration goes up, you have more carboxylase activity. So what you're doing by this sequence of events is you are increasing the CO2 concentration over what one would get in a normal mesophyll cell shown in part A of that figure, and suppressing the oxygenase activity. C4 plants have no photorespiration. I should put here Rubisco. There is no photorespiration in these plants. So they have a big advantage in that regard over C3 plants, which are losing half of their carbon as CO2. Of course, there's a problem. You know, nothing is free in life, right? In order for this cycling, this interaction between these two cell types to occur, you have to take your C3 product formed in the bundle sheath cell and move it back to the mesophyll cell. That's shown in that figure. And in addition to that, so there might be costs in terms of transporting these molecules from bundle sheath to mesophyll and mesophyll to bundle sheath. But in addition, you have to form phosphoenolpyruvate again, and that's an ATP dependent. That's a reaction that requires ATP. So what's shown in the left-hand portion of figure 133 is an ATP step in that portion of the sequence. So there's the ATP requirement for fixation of CO2 in a C4 plant is higher than the ATP requirement of a C3 plant. So you might say this is why C4 plants have not taken over the world, why most plants are C3 plants, but there is no doubt you can suppress the photorespiration activity by this particular method. It's a complicated method because you have to develop two different photosynthetic cell types. What's happening in these two cells, the bundle sheath and the mesophyll cell, in terms of biochemistry is also different. And that's not something I want to go into, but they are distinctive. They're different from each other. They're doing different things in each case. So be aware that PEP carboxylase, this pathway, in conjunction with these two different cell types, is a mechanism for suppressing the oxygenase activity of Rubisco and therefore the photorespiratory activity of the system. If you look at table 134, this is a summary of C3 and C4 plants. C3, a lot of photorespiration. C4, minimal, right? Do they both perform the Calvin cycle? Yes, both of these cells, both of these plants do the Calvin cycle, but not both cells in a C4 plant do the Calvin cycle. Primary CO2 acceptor. In a C3 plant, it is PGA, Rubisco. In a C4 plant, it is phospholynyl pyruvate carboxylase, and the product is an acid, a 4-carbon acid. That's also shown there. Class of chloroplasts, important, 1 versus 2. There are 2 different kinds of chloroplasts in a C4 plant. Okay, the last thing I want to mention is there's another system where you can suppress the photorespiration pathway. And these are in plants. Oh, if anybody wants to know what a typical C4 plant is, corn. Corn is a C4. That's the plant everybody uses for studying C4 photosynthesis, whereas in the old days, the plant that everybody used is the one you use in the lab this week, spinach. There are also plants known as CAM plants. And I'm not going to write out what CAM is because it's too long and complicated. Crustacean acid metabolism. CAM plants are, they grow in very, very hot temperatures. They're succulents. Find them in the desert, succulents. And the one that I like is pineapple. Pineapple is a CAM plant. CAM plants do something similar to this, but they do it differently. How's that for a vague statement? Okay, what CAM plants do is that they fix CO2 at night. They fix CO2 at night using PEP carboxylase. There are pores, little holes in the surface of a leaf, and this is where CO2 goes in and oxygen goes out. It's also where water goes out. CAM plants, desert plants, they can't keep their stomates, these are stomates, open during the day because they'll lose all their water. So they close their stomates during the day. Well, then how do they get CO2 to grow and to make sugars? Well, what they do is they fix CO2 at night using a similar pathway to what happens in a C4 plant. They make oxaloacetic acid or some other acid. They take this acid and they store it in the vacuole of a plant at very, very high concentrations. 200 millimolar to 1 molar. You've got this vacuole and it's full of acid. During the day, they close their stomates. The acids come out of the vacuole and are converted to a 3-carbon compound plus CO2, and the carbon is refixed again by Rubisco. So the biochemistry is pretty much the same, but what you're doing is you're separating the 2 parts of CO2 fixation that occur in a C4 plant in terms of time. So in this case, you're separating it based on structure. And a CAM plant, you're separating it temporally. I'm not sure that's a word. CO2 is actually being fixed at night in the dark in a process that does not require any photosynthetic products. And then in the light, during the light part of the day, these products formed at night, acids, are released into the same cell. There's only one cell type in a CAM plant. They're decarboxylated. Very high concentrations of CO2 are available to Rubisco, and this, again, suppresses photorespiration to a point where it simply does not exist in these plants. All right? So what we've done here is we have shown that people working in the lab trying to engineer Rubisco to get rid of photorespiration have been totally unsuccessful, but nature has used various mechanisms to overcome this problem. Okay? Are there any questions? No. Okay. Let me make a comment here. I've put up here last lecture. So this is the last lecture, right? It's really my last lecture. I've been teaching this class for 20 years, over 20 years, and I decided it's time for somebody else to step in. 8 years since I've retired, and this is it. So you guys are unique because I've tried to figure out how many students have I had in Bio 1 over the course of 20 years. It's probably, 10,000 or something, 500 a semester. But you're the last ones. I'm not like Randy, whatever his name is, the guy that wrote the book on his last lecture. He had a serious illness which ultimately killed him. That's not what is happening with me. It's just time to move on to other things. I want to just say that I've enjoyed Bio 1A as much as anything I've done at the university. I've done a lot of different things at this university. It's much more fun than some of the things that a faculty member are asked to do, and I've enjoyed you all. And all I can say is I hope you have the rest of the semester goes all right, and also that you don't suffer too much on Friday, okay? Thanks very much.